Okay, this is the last stage of photosynthesis. It's called stage 3, the Calvin cycle, or carbon dioxide fixation. Now, before we get into this, we want to review stage 1 and 2 just really briefly, because the things that are created in stage 1 and 2 will be used in stage 3. So if you'll go back and look at your notes, just this page is fine. This picture will kind of show all the things we need to look at. Sunlight hits the pigment. This is chlorophyll. The electrons jump off of chlorophyll and they are replaced by water splitting. Hydrogen ions also build up here. Oxygen's given off, and the electrons come into here and replace the ones lost by chlorophyll. So these electrons travel along the membrane, and when they pass through, electrons are energy, and so when they pass through these pumps, they make the pumps work. It's like plugging them in. So the hydrogen ions are pumped into the thylakoid from low to high concentration. That's an active transport. And a concentration gradient starts to come about because they have a lot of hydrogen coming from the splitting of water and the pumping in of hydrogen ions. And so there is a high concentration of hydrogen inside, a low concentration outside. They passively diffuse through this ATP producing carrier protein. When hydrogen moves through, this protein grabs an ADP and a P and makes ATP. So stage 1 and 2 make ATP. Really, stage 2 does. Now, electrons are also traveling through the membrane still. Light hits the pigment, excites more electrons. When electrons travel through certain proteins, they will actually take NADP plus and H plus and combine them to make NADPH in a similar way as this is happening to make ATP. So at the end of stages 1 and 2, we make NADPH and ATP. We are making those to use them in stage 3. So in stage 3, what we're going to do is similar to what we've done in stage 1 and 2. We have descriptions, and then we have a picture of the Calvin cycle, which is a circle because it's a cycle. So starting with A, carbon dioxide is added to a 5-carbon molecule. This forms a 6-carbon molecule. Carbon dioxide comes in from the atmosphere. You and I breathe it out. Plants take it in through openings called stomata that they have on the underside of their leaves. So they take it in and it enters the cycle here. So this is happening if you want to look at this chloroplast. The Calvin cycle is actually happening outside of the thylakoids but still in the chloroplast. We call it the stroma is where this occurs. So it happens outside of the actual thylakoids. Here comes the CO2 in, and like A said, one of those carbon join a 5-carbon molecule to give us a 6-carbon molecule. If you'll count those, you'll get 6. So then letter B, ATP and NADPH are used. Now where did we get those? Oh yeah, we made those in stage 2 of photosynthesis. It's called photosystem 1. We made these. We're going to use them in the Calvin cycle. So ATP and NADPH are used to split the 6-carbon molecule that we just made into 2 3-carbon molecules. So we need energy to split this up into 2 3-carbon molecules. It only shows one of these, so you can draw another one here if you like. So that's letter B. Letter C, 1 3-carbon molecule leaves the cycle. So we actually get a product out of this cycle. And we have to actually run through the cycle twice, getting 2 3-carbon molecules. And when you get 2 3-carbon molecules, you can put them together and get a 6-carbon sugar, which is glucose, C6H12O6. So let's look at that. When we left off here, we had a 6-carbon molecule, ATP and NADPH split it into 2. So here we are at letter C that says one of the 3 carbon molecules leaves. And so we see the arrow going away. It is actually leaving the cycle. It's going to find another 3 carbon molecule like I just said, and it's going to make a 6 carbon sugar, which is glucose. The other 3 carbon molecule, remember we had made 2 3 carbon molecules, the other 3 carbon molecule stays in the cycle and is changed into a 5 carbon molecule with ATP, and then it stays in the cycle to start it all over again. So we put this other 3 carbon molecule in, leave it in the cycle, and we'll label it letter D. And these descriptions here can help you as well. So the other 3 carbon molecule stays in with the help of ATP and becomes a 5 carbon molecule, and then the cycle starts all over again. You'll notice that A, we started with a 5 carbon molecule, added a carbon to make it a 6 carbon molecule. And it's a cycle, so it keeps on going, and it's self-sustaining because right over here we make 2 3 carbon molecules. One of them leaves to make glucose, the other one stays, and that's the beauty of a cycle. Something that we have to keep adding to this cycle is ATP and NADPH. We have to keep adding energy to make this cycle keep going. But the carbon that are in it are staying, some of them are staying in it and keeping the cycle going. So basically, if you sum the cycle up, the stage 3 Calvin cycle up, this is stage 3 of photosynthesis, what you get is you put into the Calvin cycle CO2, ATP, and NADPH, and what you get out is glucose. And that's basically how plants make glucose from sunlight, starting in stage 1 all the way through stage 2 to stage 3 in the Calvin cycle.